And we're perfectly timed because that leads us to the second presentation for section 2. So I'd like to now introduce Kelly to the virtual stage to present one of our newest database features, which is the custom data exports. So thank you, Kelly.
Thank you, Nina. Let me get my presentation up.
Thank you.
All right. As I said, I'm Kelly Wheeler, and I'm going to take you through the custom exports that we have. Randi briefly touched on it and showed it a little bit in her presentation, but this is the primary way that you get data out of the database when you run your queries for your particular parameters. Recently, in the last couple of years, we've updated them, and so I'm going to go through and show what the new features are for these. So previously, we had right here the export link that Randi showed, that you could choose from CSV files or CSV files with IRIs, which was a little limited on what you could exactly get out of the database and what formats to use it for. You can see here's the previous format that we had before. It had 2 header rows, you can see here, one with a section and one with a more detailed row description, and then the data below it. But again, it was only limited to CSV files and only with these dual headers, which caused some issues for some people if you're going to use them more programmatically or use them in things beyond Excel or some other visualization software. So this is why we decided to change this, because we were limited again to the comma separated ones. We only had a fixed set of export fields, which people were constantly asking for either additional fields or less fields or different fields for whatever their use cases were they had for the data to use in their own analysis of it. We had those dual headers, which caused issues from the standpoint of it was a little confusing, especially if you weren't looking at it in a more visual piece of software like Excel or something like that. We had no indications of what the fields meant exactly or examples of what it was outside of our help documentation. So you'd have to download the files and go back to the help. And so we were trying to fix that. We had no information linking what your query was to that particular file. So if you ran 3 or 4 queries back to back, downloaded all the files, you would have to mentally make note of what query was performed with those particular results. So when you went back to now analyze the data, it was a little disjointed there, which is exactly what I showed on that last one too, as well as the timing on when you ran those exports for if you archived them and wanted to run the query multiple times to see what we've added to the database between the first run of it and say the 5th run of it in a month or so. So that way we came up with these new customized database exports that allows a lot more fine tuning to your exporting and a lot more information associated with that particular export. We wanted to expand the usability of them greatly, which is the primary thing. We wanted to get more information on every field. We wanted to allow the user to customize the exports, which is a huge deal, to give you the ability to get whatever out of it that you need. And then we also wanted to provide that information linking your query to that particular data set that you exported at that particular time. So now we have the same format. You get to it from the same link that Randi showed on the tabs, and each tab has its own link. But now you can see that this pops up a new box here that you can make changes to and customize your export here. I'll go through all the different options to show you a little bit more options that you have on what you want to get out of it here. So the first off is you can now choose your format for the exports. And this was a big one because previously when we were locked into only CSVs, it's sometimes a little bit more difficult to consume those in a programmatic fashion, as well as we are adding in the ability to do the XLSX format, which is Excel's default format, which greatly expands the customability. It greatly expands the information that we can share with that, as well as being able to format it a little bit cleaner in Excel, which is where a lot of people use the data to visualize it, to do their analysis of it. Here's a couple examples of the formats. The Excel format, you can see, it's the most user-friendly. It is customized to show, to display in Excel or other spreadsheet editors in a very nice and clean structure here. The comma separated is the one that is most useful, or most standard, I guess, where it's all the data are separated by commas that you can then parse out. Tab separated is the same as the comma separated one, just using tabs instead of commas, which again, some applications prefer this format because it doesn't handle commas as well, because it can be in the data, but you can see that it is escaped in the comma separated ones. And then the last format is JSON format, which is a JavaScript object notation, which if you're a developer or a programmer, this is used widely amongst all different languages to consume data and package data into a format that's standardized. And you can see an example of it here, that you can see where it has all the information as well. This also has more data tying back to the query ran that the TSV and CSV will not. So if you want to get that information on what your query was, or when you ran it, or any of that other information, that is going to be in your Excel and your JSON formats. The next option that you can choose when you're exporting your data is how you want your header rows to be structured. In the previous version, as we showed, we only had the double headers, which is nice because it gives you sections and fields, but it isn't, some consumptions of it don't like two header rows because it expects one header row or no header rows. So those are the other options that you have. You can consolidate them down to a single header row, which basically just combines the section and the field labels into one, separated by a hyphen. And then, or if you just want the data as is, because that's how you want it, how your application wants to consume it, you can do the no header rows, which is just the raw data itself. The next option we have that you can choose here is the export type. These are pre-selected subsets of the fields that you can choose, which the IEDB team chooses to kind of group data into what we would consider maybe not our predetermined subsets, which is what we had before. But now you can choose these to quickly get a subset of data that we consider kind of the default or the basic downloads. We have the option of the full, which is all the data rows. We have the default, which is again kind of the basic one that we kind of determine is maybe not the most useful, but the most standard use there's the word I'm looking for. That we choose. We have the default, which is the same as default, but with the IRI, which are the identifier columns, which is useful for if you want to join those data or get the data, get the identifiers for those particular pieces of data. Say an antigen, you want the UniProt ID, that would be stored as an IRI column, which would be included in the default with the IRIs. And then you have the IEDB website displayed, which is a set of columns that are match exactly what you see on those tabs when you export, which we also did not have previously in our exports, was the ability to get exactly what you see on those tabs, which we had people asking for, because that gets some of the counts data and some of the other data that we just didn't have it able to do it for. And then you have the custom export, which is the real meat and potatoes of this new export format, which allows a user to choose exactly what they want. So here's a little bit more on the export, the IEDB website displayed export type, because it's a special one. Because of its specialization, you cannot modify the fields that are chosen here. All the other ones you can choose, pick and choose your fields, but if you want the IEDB website displayed, you get basically what is displayed there and shown, but this is how you can get exactly what is shown on your results page if you want those. So now I'm going to go into the custom export option, which is really where the most of the power in this custom exporting lies. It allows you the fine-grained customization to pick and choose every field that you want or don't need from your exports. You can see it's shown in kind of a tree structure here, where the section headers are shown above, and then the individual the fields are below those. You can pick and choose any of the fields that you want in here, and you can add or remove entire sections or fields by just clicking on the box next to it. As you can see in this here, if it's a full green box, you're selecting everything underneath that. If it's a half green box, it means you've selected fields underneath that. So when they're collapsed, you can see, okay, I have, one or more of the epitope fields selected. But up here, you can see that you have none of it. If there's no green in there, then you have not selected that section at all. So here's some examples of the different tabs that we have the exports for. You have the epitope tab, and there's more options in these. There's just, as you can see by the scroll bar here, you have the antigen tab, which this one is very basic for right now. We haven't expanded it. It is basically the website displayed, because we haven't expanded that out fully. You have the assay tab, which is each assay is going to have its individual set of fields and that you can choose. And as you can see, there's a lot of them in here, because this is where we have a lot of our data is associated with the individual assays. And like I said, each assay will have its own subset of sections and fields to go along with it. And then you have the receptors, which you can see they have all their different fields as well. And then the reference, which is, again, very basic, but it expands out some from the website displayed as well. Now, on top of the ability to customize your exports, which I explained, there's now inclusion of the query parameters in the Excel and the JSON exports. So you can go back and see exactly what you queried on in your previous query for that data set. You also have the export date in the Excel and the JSON exports. So you can, again, so if you regularly visit the IEDB and once you see what data we've added between two time periods, you can quickly tell, okay, this one was ran on this date and this one was ran, a month later or something. And then also we have the field descriptions in the Excel export via new sheets, which shows the real power of including those Excel formats rather than just the TSV or CSV formats, that they can have the additional sheets which show you a lot more data, which I'll show you here in the next couple slides. So here you can see the export parameters, which should match exactly what you see on in the filters section on the website. So this example here, you can see that it was, linear sequence. We searched for this particular linear sequence, just positive assays, and then you can see what type of export type, if you selected one, is here. And then as well as the date exported, you can see down there at the bottom. These are available again in the Excel and in the JSON. And you saw earlier how it's expressed in the JSON format. It's another section there that can be consumed. Okay, then the other tab we have, and the other sheet we have in the Excel formats is this field help section, which explains all the fields. Every field that you selected will have this little demo here, or this little help section here that is broken down exactly like you see before. You have the section, you have the headers, and then below that you have two examples that we've chosen to show just examples of one of those rows. So you can see that here, and then below that is just another possible exception that will show everything in case the examples that we chose or your fields that you chose don't have any data. So like you can see here, the modified residues in these examples aren't shown, but there is data here that can be possible there, and this shows an example of what that could be. Below that, we have a description of the field, which is just a brief text description of what that represents. And you can see that here, this is the URL for the epitope on the IEDB site. And then you can see here. And then below that, if we have a help link, then we have that below that. And then finally, some of the improvements that we are looking to bring to these going forward in the near future is these full custom exports. We are adding them soon to the query API for the IEDB, which I'll be speaking on actually on day 3. I'll explain that a little bit more. But the full exports are going to be available there. We are exploring including the API URL to be able to reproduce your query through the API and incorporating that into the IEDB website and the custom exports. And then we're also evaluating all the available fields and export types continuously. So if we need to add more or modify those types, then that'll be done on the fly as the team determines it. And then also our sister site, Cedar, we are looking at customizing the exports there to have Cedar-specific information. So it's more specialized for the cancer data. I think that's, am I my time, Nina?
That is perfect timing. Thank you so much, Kelly. Much appreciated.
Thank you.
Perfect. So I'll give you a couple of minutes to take a look at the chat, not the chat, excuse me, the Q&A, as we do have some questions coming through. So now I'd like to invite Randy back to the Zoom, and with Kelly, we'll start section 2 Q&A. So Randy, take it away with the first question.
Okay, let's see. Once I said it was, oh, sorry. So one person had asked, anonymous attendee, when trying to merge downloaded CSV files, such as epitope, antigen, receptor, and assay, which ID column should I use to join that data together? So the epitope ID will be in all of those exports, but just know that the antigen will have many epitope IDs from it, and the receptors can bind also frequently to many different epitope IDs, but each epitope will have one or more assays, and an antigen if it's natural. And then also, if you're savvy, you can download the entire database. You can download it in SQL format and perform other queries. And one of the other questions that Kelly alluded to is someone had asked if we have an API. And yes, that Kelly is going to talk about that on the third day. So, Kelly, did you find any you wanted to?
Yeah, I can
ahead.
take, somebody had a question about timing out. It should not time out. Just some queries may take longer than others, but they should return back. So there shouldn't be a timeout once you start using it. You should be able to keep running that continuously.
Okay, and I'm going to take a question from anonymous attendee asking, what is an IRI? That's a very good question. It is a stable identifier for a term. And so if it's an ontology IRI, I'll say it'll be a disease ontology identifier. It just basically means an ID, and it's a stable ID, and they'll link out, there'll be URLs, and you can go to that ontology's website and read more about that assay type or that disease. Or for NCBI taxonomy, it'll take you to an NCBI taxonomy page for that organism. We also have internal IDs for each epitope, assay, and reference and receptor. Okay, go ahead, Kelly.
I had an anonymous attendee asking about exporting large data. It shouldn't, I don't know how exactly you would assure that nothing went wrong, but it shouldn't go wrong. If it's a large amount of data, it will still run. And let me pull up, actually, I have a page here. Let me share my. So when you download, when you run a custom export, it will link you to this page here, and this will say when it's running that it's downloading, and it will notify you when it's complete here. And 24 hours after it's complete, it'll still be there. So you can leave this page up or bookmark this page and come back to it, and it should still be there. So if you want to run one that's particularly taking a little while, you can come back to it, in a couple hours and see if it's done. It'll also automatically update it and try to download it for you once it's done. So you should be okay. It should still work, and it will zip it up if it's particularly large, or if it's a CSV, TSV, or I think JSON files actually zip as well to save time and space.
Okay.
Do you want to take the next one, Randy?
Yep. I'm going to take anonymous attendee asked, what if someone is interested in normal samples, no disease? Can you search for that? Yes, you can. You use the disease search box, and healthy is one of the options. You can, or if you've done any query like I did for all influenza epitopes, when you look at the assays, there's a column that tells you whether the host had a disease or did not have a disease, the disease state column. So either you can search specifically for it, or you can see it in your results. And Nina, I wanted to point out that there's 2 questions about binding prediction, and I think we should save those for the prediction people, to set them aside for when we're doing the tools talks.
That sounds good. Thanks for bringing that up, Randy, because there were some questions from the first section Q&A that Bjorn did put aside for tomorrow as well, because it will just make a lot more sense in the context of tomorrow's presentations. So for those of you who have asked about binding or epitope prediction related things, can you please post them in the Q&A tomorrow, and then we will go through them in a lot more detail. Thank you. Thanks, Randy.
And yeah, Kelly still has some more.
Yeah, I'll take the next couple. As far as browsers, we we officially support Chrome, Firefox, and Edge, but all of them should work fine. There's not going to be any kind of any differences really in the compatibility. As far as how long you should wait, they generally don't take more than they're generally really quick to generate the exports. If it doesn't work after, a couple hours, maybe shoot us off a message through the help center, but it should work after a couple hours at the very longest. And at that point, you're almost better off to get the whole data set and do your own filtering on it, because in order for that to take that long, you're generating probably the majority of the database anyway. So you're not filtering out a whole lot. And I will say, if you run one of the default queries or the full database queries, it will be pretty quick, because the system is set up to give you the full data set or pre-canned ones that we pre-generate those when we generate the database to speed up that process and not have to wait multiple hours.
And Nina, I found one question in the chat that I would like to answer. I'm sorry, I can't read the name. It starts with MAIR. I was asked, what does negative data stand for and how important are they? And this is the last one I have to answer. So when any experiment is done, the authors have to let us know if they considered it positive or negative. Was IL-2 produced in this context or not? And we go entirely with what the authors say. And negative data lets you understand in what context it was negative. So for example, an epitope may give a response in an allergic subject, but not a healthy subject, or a patient with autoimmune disease, but not in a healthy patient, or not in a patient with a different autoimmune disease. Also, different strains of mice, based on their MHC restriction, they may get a response or not. And so that may be interesting to you to know that it works in a BOBC mouse and not a Black 6. So you could tailor your experiments that way. So the negative data, the context is entirely up to the users and what's relevant to them. Okay, so that's it for me. So Nina, take it away.
Excellent. Thank you so much, Randy. Thank you, Kelly, for going into the database and the exports in a lot of detail and answering those questions. So we are running a little bit ahead of schedule, about 8 minutes or so. So that just means we'll have a slightly longer break. We were going to have about a 15-minute break. It'll be about 22 minutes or so. So stretch your legs, get something to eat, and then meet us back at 10:40 a.m. Pacific time, which is in about 20 or so minutes. We will post some resources and the timing in the chat as well. And I'll play some videos during the break. If you happen to be around in the break, they'll be playing on the screen, and then we'll make sure to link you to them on our YouTube as well. Continue to post on Twitter or X for us, which we've seen a few already, so thank you so much. And we will continue to do the same. So with that, everyone, thank you so much for the first 2 sections of day 1. We'll come back in 20 minutes and go through section 3.